Fate, mapping and lineage tracing in embryonic development. Fate mapping and lineage tracing are powerful techniques used to study the early development of embryos, helping to understand how specific cells contribute to the formation of tissues, organs, and structures in the developing organism. These methods are essential for uncovering the mechanisms of cell fate, determination, differentiation, and morphogenesis during embryogenesis. First, fate mapping, definition and methods. Fate mapping refers to the process of tracking and identifying the future developmental fate of a specific group of cells in an embryo. It aims to determine which tissues and organs specific cells will contribute to as development progresses. Fate mapping is essential for understanding how different parts of an embryo form, where particular cell types arise from, and how the germ layers give rise to various tissues. Techniques in fate mapping. Vital dyes. One of the earliest methods used for fate mapping involves labeling specific cells with vital dyes, which are non-toxic and can be detected under a microscope. The labeled cells are tracked throughout development to observe their contributions to various tissues and organs. Cellular markers and genetic labeling. Fluorescent proteins, e.g., GFP or RFP, can be used to tag specific cells. These proteins are expressed in certain cells, and their fate can be tracked over time. The fluorescence allows researchers to visualize the location and movement of labeled cells during development. Genetic labeling involves inserting a marker gene into a specific cell population. This allows the tracking of cell derivatives without affecting the normal development of the cells. In situ hybridization, this method involves detecting specific mRNA sequences in cells to identify gene expression patterns and predict the fate of certain cell populations. It is especially useful for examining how signaling molecules influence the fate of specific cells. Chimeric embryos. A chimeric embryo is created by combining cells from different embryos. This technique is often used to study the fate of specific cell populations by observing how transplanted cells contribute to the development of tissues in a new host embryo. Second, lineage tracing, definition, and methods. Lineage tracing is a technique used to follow the ancestry and developmental history of individual cells or groups of cells from their origin during gastrulation to their final differentiated state in a mature organism. Lineage tracing helps to understand how single cells give rise to multiple cell types and contributes to clonal analysis. Techniques in lineage tracing, genetic labeling, and inducible systems. Cree-Logsby system. This is one of the most commonly used methods for lineage tracing. In this system, a Cree recombinase enzyme is used to activate or deactivate a marker gene in a specific population of cells. This allows researchers to track the descendants of those cells as they differentiate into various tissues. Inducible systems like TET-on/TET-off can be used to control when the lineage tracing marker is activated, allowing precise control over cell labeling at specific developmental stages. Fluorescent protein labeling. Using fluorescent proteins like GFP or RFP, specific cells can be labeled and tracked over time to observe their migration, division, and differentiation. This method is especially useful for studying how cells contribute to tissues and organs over time. Single-cell RNA sequencing. This technique allows for the analysis of the gene expression profiles of individual cells. By performing RNA sequencing at different time points, researchers can track how the expression of specific genes changes in a lineage, helping to identify the developmental pathways that a specific cell population follows. Transgenic mouse models. Transgenic mice expressing reported genes, such as GFP, under the control of specific promoter sequences, can be used to track the fate of specific cell populations throughout development. These models provide detailed information about the spatial and temporal dynamics of cell differentiation. 3. Applications of fate mapping and lineage tracing. A understanding early embryonic development. Fate mapping and lineage tracing are fundamental tools for understanding the process of gastrulation, neurulation, and other early stages of development. By tracking cell fates, researchers can determine which regions of the embryo give rise to specific organs, tissues, or structures. B. Studying cell differentiation. These techniques allow scientists to investigate how stem cells differentiate into various cell types, helping to unravel the process of stem cell differentiation during development and tissue formation. C. Identifying developmental pathways. Fate mapping and lineage tracing provide valuable insights into signaling pathways, e.g., BABWINT, BMP, Notch, and Hedgehog, that regulate cell fate determination. By tracking cell lineages, researchers can correlate specific signaling pathways with tissue formation. D. Studying regeneration. These techniques are also used in regenerative biology to understand how some organisms, like salamanders and zebrafish, can regenerate lost tissues. By tracking the lineage of regenerating cells, researchers can identify the molecular signals responsible for this process. E. Investigating developmental diseases. Lineage tracing helps researchers understand how developmental defects arise, such as those seen in congenital disorders, cancers, and neurodevelopmental diseases. By tracing the developmental history of diseased cells, scientists can uncover potential causes of abnormal tissue development. Fourth, challenges in fate mapping and lineage tracing. Despite their many advantages, these techniques come with certain challenges. Resolution: tracking individual cells over time, especially in large or complex tissues, can be difficult. Advances in high-resolution imaging and single-cell techniques are helping overcome these limitations. Labeling efficiency. In some cases, the efficiency of labeling specific cells can be low, and there is a risk of loss of labeling during cell division. Efforts to improve genetic labeling strategies are ongoing. Ethical considerations. Some techniques, especially those involving genetic manipulation or creating chimeric embryos, may raise ethical concerns, particularly in human studies. Careful ethical review is necessary when using these techniques in research. 5. Summary. Fate mapping and lineage tracing are invaluable tools for understanding the development of the embryo and the differentiation of cells into specific tissues and organs. These techniques have wide-ranging applications in developmental biology, stem cell research, and regenerative medicine, providing insights into the mechanisms that govern cell fate determination and organ formation. While challenges exist, advancements in these technologies continue to enhance our understanding of embryonic development and disease processes.